Muscle structure and organization. We're going to begin looking at skeletal muscle tissue. Some of the general characteristics of skeletal muscle and tissue include that it is striated. When you look at it underneath the microscope, you're going to see a distinctive banding pattern running through the cells. It's also multinucleate. That means that there's more than one nucleus per cell. Skeletal muscle tissue is also voluntary. If you make the decision to move a skeletal muscle, it will in fact contract. It's not something that happens automatically. Skeletal muscle function. There are a couple different functions that fall underneath skeletal muscle. One of them is movement. Clearly, skeletal muscles, by their name, are attached to the skeleton, so some muscles would be like the biceps brachii or the gluteus maximus. Those are some common skeletal muscles. They attach to bone, and when that muscle contracts, they allow for movement. Muscles can be synergistic. What that means is that they work together to form one action. For example, if you know where your biceps brachii is, there's a muscle just deep to that or underneath it called the brachialis. When those muscles contract, they both flex the forearm. You'll see the same thing with your quadriceps muscles on the front of your thigh and your hamstring muscles on the back of your thigh. So by definition, synergistic muscles are muscles working together to create the same movement. Muscles, however, can also be antagonistic. That means that they're going to be working against one another. For example, if we use the biceps brachii again on the front of the arm, the antagonistic muscle to that would be the triceps brachii on the posterior side of the arm. So again, antagonistic muscles are simply muscles that oppose each other's actions. When we look at the motion or the action of a muscle, we need to talk about two terms. One is called the origin. The origin is the part of the muscle that is the least movable. It's the fixed end. Usually, the origin is going to be the most superior portion or the most medial portion. Regardless, it's where the muscle begins. The insertion then would be where the muscle ends, and it ends up being the movable end, or the end of the muscle that's more inferior or more lateral. Another function of muscle tissue is generation of heat. And if you don't believe me, just get up and start running around. The more you run, the more heat you're going to produce because you're using your muscles. Taking a look at this picture, you can see that in picture A, there are antagonistic muscles. There's the biceps brachii on the right, or the front of the arm, and there's the triceps brachii in the posterior, or the back of the arm. In the picture, you can see that as the biceps contracts, the forearm gets flexed. As the triceps brachii contracts, the forearm gets extended. We're now going to look at the organization of muscle. We're going to begin with the largest and move down to the smallest. So the largest organization is, of course, the muscle itself. This is if I were, to get my scalpel out and dissect you, I'd be able to pull out individual muscles in your body and hold them in my hands. So the muscle is the whole structure. You are responsible to identify the skeletal muscles in your text on Figure 6.2 on an exam or any other assessment tool. This should be done on your own. If you have questions or concerns, again, by all means, let me know, but I would suggest, either covering them up again with Post-it notes and studying that way, or you can go to the textbook website and they have activities that will allow you to label the muscles as well. Now, if we go from a muscle to the next smallest layer of a muscle, we're going to run into what we call fascicles. Fascicles are bundles of muscle fibers, or in other words, bundles of muscle cells. So, the easiest way that I can explain this is if you've ever made a roast at home, you take the roast, which is, the entire muscle, you put it in, say, your crock pot, cook it all day long, and when you go back to your roast, the muscle falls apart into nice little bundles. Those little bundles are fascicles. If we went a little bit deeper to a smaller layer of a muscle, we'd find muscle fibers. By definition, muscle fibers are cells. So this layer you're not going to be able to see with the unaided eye. You are going to need to have a microscope to see these. They contain organelles called myofibrils. if you take a look at this picture, what we have is the muscle, that's the largest structure, and then you can see they've taken a cross-section of the muscle and they've pulled out one of the bundles in there. That bundle is a fascicle, and then you can see multiple fascicles, of course, inside of that muscle. They went a little bit further then and pulled out a muscle fiber or a muscle cell from the fascicle. So you can see that there are multiple layers of organization to a skeletal muscle. Myofibrils. These are organelles, like we had mentioned before. They are surrounded by the plasma membrane of the skeletal muscle cell. And what happens is that the plasma membrane continues along and dips down in between the myofibrils. So transverse tubules, or T-tubules, are extensions of the plasma membrane that plunge down between adjacent myofibrils. Their function is to transmit an electrical impulse. Sarcoplasm, that might sound a little familiar to you because it sounds a little bit like cytoplasm. Anytime you see the prefix sarco, think muscle. So sarcoplasm is going to be the cytoplasm of the muscle cell, and it is in fact surrounding the organelles or the myofibrils. There's another structure inside of the muscle cell called the sarcoplasmic reticulum. The sarcoplasmic reticulum, as you might think, sounds a little bit like endoplasmic reticulum. So it's similar to the smooth endoplasmic reticulum. However, the difference is that its function is going to be to sequester calcium. That means to hold calcium inside. Taking a look at this picture, at the top we've got one muscle fiber or one muscle cell. You can see that the plasma membrane has been pulled back a little bit, and underneath that at the top you can see the two purple nuclei. And then they've pulled out a myofibril. So if you're looking at one muscle cell as the entire thing, a bunch of myofibrils are packed inside. And then on the bottom what you see is the actual image you would see through a microscope if you were looking at this same thing. This schematic as well shows you some myofibrils. They're the circular structures, and you can see around the outside the light pink or peach colored continuous sheet would be the sarcolemma. That's going to then have some extensions that dip down in between adjacent myofibrils, and you can see that that's that white tube traveling with the little yellow arrow through it. On either side of the T-tubules, you will see that there's some blue tissue. That is the sarcoplasmic reticulum, again, whose function is to sequester calcium. Now, if we want to look at the functional unit of a muscle cell, we would call that a sarcomere. This is what's actually doing the contracting inside of the muscle. If we wanted to put borders on it, we would say it runs from something called a Z-line to the next Z-line. And inside we have a couple proteins. The first protein is something called actin. If you were to look at actin structurally, it kind of looks like a twisted strand of pearls, or maybe like a twisted strand of Mardi Gras beads. And attached to the actin, we have a protein called tropomyosin. The function of tropomyosin is to block the myosin binding sites on actin. So at rest, a muscle cannot contract because the tropomyosin is blocking the binding sites. Now, attached to the tropomyosin, we have another protein called troponin. Troponin sits on top of the tropomyosin, which sits on top of the actin. And troponin has a binding site for calcium. So somewhere in this mechanism of action, calcium will bind to troponin and expose the binding sites. If we take a look, this is a sarcomere, and you can see that the sarcomere runs from Z-line to Z-line. They've blown up one sarcomere. You can see that the light gray structure is actin, or a twisted strand of pearls as it appears, and the orange structure in the center is the myosin. This picture shows you what happens when the muscle is relaxed. So the top picture is when the muscle is relaxed. You can see that the myosin is not holding on to the actin at all, and that there's quite a bit of distance between the two Z-lines. In the bottom picture, however, that shows how the muscle actually contracts. When the myosin heads grab onto that actin, they kind of pull the sarcomere towards the center, and the distance between the two Z-lines gets shorter. Myosin then is one of the other contractile proteins that I've been mentioning. This kind of looks like a bundle of golf clubs. They have heads, which we'll call cross bridges once it grabs onto the actin, and they also have a tail portion that's kind of anchored in the center of the sarcomere. The sliding filament theory is the theory by which a muscle contracts. What it means is that the filaments, both the actin and the myosin, when contracting, simply slide past one another so that the muscle shortens. I put on here a link to a video for you to watch. Please watch the following video. I have a little note that you do not need to know about the different lines and zones inside the sarcomere. I simply want you to see the process by which the sliding filament theory takes place. So go ahead and check that out and see if it doesn't help clarify this for you. Again, this picture is just going to show the difference between the relaxed state on top or the contracted state on the bottom. And again, you can see that on the bottom where the muscle is actually contract, the actin and myosin filaments have slid past one another to shorten up that sarcomere.